Hello everyone, my name is Natalia and in this video I'm going to present my PhD project entitled Epidemiological Aspects of the Microbial Structure and the Gut Resistome of Children in a Watershed Area in the One Health context. My advisor is Professor Ana Lúcia da Silva and co-advisors Cláudio Galupo, Vanessa Dias and Francisco Teixeira from UFRJ. The indiscriminate use of xenobiotics such as antimicrobial drugs, biocides, and toxic metals has resulted in the emergence of multi-drug-resistant bacteria which no longer respond to traditional therapies. One Health is the integration between human, animal, and environmental health. It's an approach to designing and implementing programs, policies, legislation, and research in which multiple sectors communicate and work together to achieve better public health outcomes. Although antimicrobial resistance is a natural phenomenon as a result of natural selection, anthropogenic activities have contributed to the increase of its occurrence. Bacteria can share resistance genes in the gut. However, little is known about the presence of resistance genes in the gut, known as the gut resistome, especially in the absence of selective pressure attributed to antibiotics. Therefore, the main goal of this study is to evaluate the occurrence of antimicrobial resistance genetic markers representative of the human gut resistome in fecal specimens obtained from school-aged children living in the watershed area of Rio das Ostras, Rio de Janeiro. To achieve this goal, representative DNAs of the gut metagenome will be obtained from fecal specimens of children aged 6 to 10 years old, categorized into students from predominantly rural or urban areas. Afterwards, the aspects of the gut microbiota structure of the children sample will be evaluated by DNA fingerprint technique, from which a dendrogram will be constructed to analyze the clusters. The presence of antimicrobial resistance genetic markers from the gut resistome will be evaluated by PCR. The occurrence and distribution of antibiotic resistance markers will be correlated with the clinical and nutritional data of the children sampled, and the gut resistome of the children evaluated will also be correlated with the occurrence and distribution of antibiotic resistance markers along the course of Rio das Ostras, considering areas with different levels of urbanization. For this purpose, statistical analysis will be carried out. These are the references used in this presentation. Thank you for your attention.